Designing a simple experiment. To illustrate general concepts underlining experimental design, let's work through a simple illustrative experiment. This experiment will study the effect of usable soil nitrogen on corn productivity. The elements of a controlled experiment that are explained on the following slides will prepare you for the labs in the remainder of the course. All scientific inquiries begin with a scientific question. The source of the question can be from reading, an observation, your imagination, etc. So the question here, does the amount of usable nitrogen in the soil affect the number of seeds produced by corn plants? Hypothesis, the amount of nitrogen in soil affects the production of corn seeds. Scientific approach, plant many corn seeds in soil with varying amounts of usable nitrogen and count the seeds produced by the plants in the different soils. In this experiment, which is the independent variable, which is the dependent variable, and which unintended variables may be difficult to control and could impact the data? The independent variable, that which the experimenter is varying throughout, is the nitrogen in each soil sample. So that's changing. We have changing amounts of nitrogen in each soil sample. The dependent variable is the number of seeds produced. So what we're measuring is the response to the nitrogen in each soil sample by the number of seeds produced. Which unintended variables may be difficult to control and could impact the data? Here are some possible answers. The amount of nitrogen already in the soil, the types of bacteria in the soil, variation in the quality of the seeds used, slightly different growing conditions, variation between sources of usable nitrogen added. Background research. Background research is critically important since a key to the success of science is that each new experiment builds on prior ones. The first step in designing any experiment is to research what has been learned previously. Some facts pertinent to our experiment. Commercial fertilizers have mixtures of these usable nitrogen sources, urea, ammonia, ammonium, and nitrate, and they vary. Usable nitrogen content has been measured in the soils of many corn farms and found to vary from 100 to 450 parts per million. In one study, 400 parts per million gave the best yield of corn seeds, although higher concentrations were not tested. The soil of our test field has a usable nitrogen content of 100 parts per million. Designing a simple experiment, positive controls and negative controls. A positive control sample has all the necessary variables and conditions required for a measurable change to occur. It should work well and give a strong result. If it fails to produce a result, the system is not working. A negative control sample lacks a crucial element required for a measurable change to occur, so there should be no measurable result. It's a sample that should not work. A measurable change in a negative control would indicate that there are unintended variables impacting the experiment. The results are erroneous, and the experimental design should be reviewed. Based on our background research, approach, and hypothesis, design a positive and a negative control for this experiment. Possible answers include, for a positive control, add 300 parts per million usable nitrogen to the soil of some plants, bringing that soil's nitrogen content to 400 parts per million. As a negative control, add no nitrogen fertilizer. This should eliminate gain relative to soils to which nitrogen is added. Another negative control, remove all nitrogen from the soil or block the uptake of nitrogen by seedlings. There should be no growth and no seeds produced. If plants grow, then there's an unidentified source of usable nitrogen that will impact results. Designing a method. Choosing a method that will not introduce unintended bias requires attention to details. A soil nitrogen test showed that the experimental field had very low concentrations of soil nitrogen, 100 parts per million. In previous experiments carried out in a different part of the world, the ideal total nitrogen content was 400 parts per million. So, the field was divided into 24 strips of equal area. Each strip would receive 0, 50, 100, 150, 200, 250, 300, or 350 parts per million of nitrogen added to the existing 100 parts per million in the form of a commercial fertilizer. Corn seeds were planted and plants were grown to maturity. Collecting and analyzing data. Random sampling of a population, calculating average values, and representing data in tables and graphs are all elements of analyzing data. Corn plants were selected randomly from each strip and their corn seeds or kernels were counted. The average number of corn seeds per plant was calculated for each concentration of nitrogen. Raw data was organized in a table. Describe a corn plant sampling method that would minimize bias in data collection. One way would be to assign a number to each plant or group of plants and randomly select the numbers of the plants to be studied. That would avoid sampling bias that could be introduced by having a person pick out the plants to study. They could consciously or unconsciously pick out plants that would support a certain outcome. Also, it would be better than picking every other or every third plant, since that could lead to a correlation between the plants chosen and their location in the field. That would introduce sampling error. So I'm showing in the table the total nitrogen in parts per million from 1 to 450 and the corresponding average number of seeds per plant. And each of these values, 1.1, 2.2, 3.5, and so on, are measured times 10 to the minus 4. So 1.1 times 10 to the minus 4, 3.9 times 10 to the minus 4, and so on. So let's plot these points to see if there's a relationship between the variables. Then decide the type of graph to draw based on those results. After that, let's hand draw the graph. An optional graphing practice, use a spreadsheet to create a graph. As a review for choosing the type of graph, scatter plot for determining the mathematical relationship between two variables by drawing a line of best fit. A line graph is for analyzing a relationship between variables that may not be mathematically simple or may not extend over the range of values in the graph. A histogram is for comparing data points that are continuous and very numerous. The data must be divided into intervals. And a bar graph is for comparing discrete groups of data. Choose the best type of graph for the corn seed yield versus usable nitrogen data before going on to the next slide. I've shown on a scatter plot here total soil usable nitrogen in parts per million on the x-axis and average seeds per plant on the y-axis. These points could justify using a scatter plot with a trend line or a line graph. Let's look at each and make a judgment as to what we learn from each. A scatter plot yields a reasonable looking linear correlation between the variables. However, the points deviate from the trend line when above 350 parts per million of nitrogen. That could reveal a limit to the benefits of added nitrogen, or it could be due to inaccuracy of the data. Let's next look at this as a line graph. The line graph supports the idea that adding more than 350 parts per million of nitrogen does not increase the yield, as we can see the graph level off here. Between 100 and 350 parts per million, there is a positive correlation, but beyond that, the effect seems to level off. If the data had included information about the accuracy of the measurements, we could better determine if this is a real effect. Subsequent experiments beyond 450 parts per million would confirm if the leveling off is real or due to measurement error. Calculate the gain in yield or seed production per 1 part per million usable nitrogen added based on the trend line given. This will be the rate at which the yield changes per unit of nitrogen added. This is found from the slope of the trend line, not from the points. The orange triangle shows that the rate or the slope is equal to the change in y over the change in x. So I have a point up here at 6.0 times 10 to the minus fourth minus 2.0 times 10 to the minus fourth number of seeds divided by what is x here. X here is 375 minus x here is 125 parts per million of usable nitrogen. That's 4 times 10 to the fourth seeds per 250 parts per million of nitrogen, which is a rate of 160 seeds per parts per million nitrogen. What might yield a more accurate result for the range between 200 and 350 parts per million? A more accurate rate for the range from 100 to 350 parts per million nitrogen might be found by drawing a trend line that just omits the points beyond that, where we saw a leveling off revealed by the line graph. So we can find the slope of this trend line using the orange triangle to find the rate, the change in y divided by the change in x. So my y up here is 6.25 times 10 to the minus 10 to the positive fourth. And my y here is 1.0 times 10 to the fourth. So the change there is 5.25 times 10 to the fourth seeds. And my change in x is from 385 parts per million to 100 parts per million, or a change of 285 parts per million of nitrogen. And our rate here is for each additional part per million of nitrogen, we get an increase of 185 seeds. This is a greater rate than we calculated on the previous slide before reaching the 350 parts per million of nitrogen, before that leveling happens off. Claim, evidence, reasoning. AP Biology curriculum favors the claim, evidence, and reasoning approach to developing and justifying scientific arguments. Claim is a conclusion based on the experiment. Evidence is the experimental data that supports your claim. And reasoning is an explanation of why the data are valid evidence for the claim and why the claim is valid based on the data. Based on our model experiment, make a claim about the results, cite evidence that supports your claim, and demonstrate your reasoning for making that claim and choosing that evidence. Claim. Adding nitrogen to bring soil nitrogen levels to 350 parts per million is correlated with increased productivity of corn seeds. Evidence. We measured a positive correlation between corn seeds and nitrogen over a range of 100 to 350 parts per million. Reasoning. Increasing doses of usable soil nitrogen while keeping all other variables constant is a valid approach to testing the effect of usable nitrogen on corn seeds. Counting seeds is a direct measurement that allows no significant error if sampling is without bias. Results showed that with each increase in nitrogen, we also measured an increase in production of corn seeds, and so the evidence strongly supports the claim and the hypothesis. New questions, next experiments. Every experiment leads to new questions. 1. Pose new scientific questions based on the experiment completed. What are you wondering about? Can you turn that curiosity into a specific question? 2. In one sentence, describe the next experiment you would do to further your knowledge in this general area. After thinking about these challenges, see the sample answers on the next slide. Possible new questions. 1. Does corn seed production continue to increase with more fertilizer, or does it level off above 350 parts per million? 2. At what concentration does excess nitrogen become detrimental to seed production? 3. How do other crops respond to increased nitrogen? 4. Would the results for corn change if other variables changed? For instance, if it were hotter, wetter, sunnier, etc. Possible experiments which correspond to those questions. 1. Repeat the experiment with higher levels of nitrogen in similar fields to see if yields really do level off above 350 parts per million. 2. The same as number 1. In this case, to see if the yield versus nitrogen takes on a negative slope. 3. Repeat the original experiment with other crops. 4. Repeat the original experiment, changing one of the indicated variables, temperature, moisture, sunlight, etc.